Hello students, welcome to EPG Pathshala. I am Dr. Ashish Pal, scientist from Institute of Nanoscience and Technology, Mohali, Punjab. Today we are going to discuss a module on understanding the structures and interactions in nucleic acid under the paper Nanobiotechnology. After completing the module, you should be able to understand what are the difference of nucleic acid building blocks. What are the types of nucleic acid building blocks available? Primary and secondary structures of different nucleic acid like DNA or RNA, and how do they form, and where are the potential binding sites of in the nucleic acid where other ligands or other material can bind. So this knowledge of the interactions between these nucleic acid is very important, because eventually it will help us in understanding the strategy to form nanostructures mediated by the nucleic acids. Just after the discovery of DNA double helix, Richard Feynman in 1959, in his lecture in Caltech, he actually envisioned that DNA can be used as a storage material, but it was too early for that time to understand the implications of his comments. Now, when we look at DNA in the present world, we do see, yes, DNA is a molecule, and it functions successfully as a genetic material because of its chemical property. It has a very nice complementary sequence, and it has anti-parallel double helical backbone, and it can actually read the sequence. Well, now DNA is basically any nucleic acid, if you take, that can be of two types. One can be deoxyribonucleic acid, that is DNA, or ribonucleic acid, that means RNA. Now, there are minute difference, we will come to that there, but I think what I want to say, for simplifying to the students, DNA is just like Coca-Cola. In Coca-Cola, what you have sugar, like, okay, you also have phosphoric acid, and you also have caffeine as a base. Similarly, in DNA, what you have phosphate group present in there. You do have the sugar unit, which can be deoxyribose in case of DNA, or we'll come to RNA that it is ribose. And there, instead of caffeine in Coca-Cola, here you have the nucleobases. So let's look at the the different fragments of DNA, or the basic building block of DNA. The nucleic acids are nothing but they are polynucleotides, and if you look at one particular nucleotide, they are made up of nucleoside and phosphate, and nucleoside is made up of those bases which can be of two different type, purine or pyrimidine, and sugar can be different for DNA and RNA. Let's look at the structure of the DNA. There are two types of nucleobases. One type is purine, under which there would be uracil, thymine and cytosine. Thymine and cytosine one can find in DNA, but in RNA, in place of thymine, we get to see uracil. The other type of base, nucleobase, which is present in DNA or RNA, is pyrimidine base. And there are two types of pyrimidine bases. One is adenine, another one is guanine. Now, these nucleobases, when they get combined with the sugar unit, then it is called nucleosides. Now, there are, depending on DNA or RNA, the sugar units are different. In the case of DNA, the sugar unit is deoxyribose sugar, where if you look at the structure of the sugar, on the second carbon the hydroxyl group is missing. But in the case of RNA, the sugar backbone, that is consists of ribose, and second position of the carbon has hydroxyl group in it. And now these nucleosides comprising of the nucleobase and the sugar unit, when that gets coupled with phosphate group, so then we form called nucleotide. Now, when you have a mononucleotide, that means you are talking about one phosphate group, one ribose and nucleobase. Now that consists of a mononucleotide. Now couple of mononucleotide, when they get linked up, then they can form oligonucleotide, and eventually when you have more and more number of such mononucleotide forming a chain, it can have polynucleotide. Eventually that polynucleotide is something which we called nucleic acid. If you look at the sugar unit for deoxyribose for DNA and for RNA it is ribose, the first important thing, as we have already mentioned, that second carbon atom, in case of DNA it doesn't have a hydroxyl group, but in the case of RNA it has a hydroxyl group. Now presence of this hydroxyl group makes it very different character for RNA and DNA towards alkaline hydrolysis, because in RNA the RNA backbone that is more susceptible to towards alkaline hydrolysis because of presence of this neighboring hydroxyl group. Also another thing is to be noted, the conformation of this ribose and deoxyribose unit may be different. You can see here in the ball and stick model, in the case of deoxyribose and ribose sugar, they are little different. Also the hydrogen bonding involving the second hydroxyl group also stabilizes some of the RNA structure. We can see some of the secondary structures of RNA. So this shows the primary structures of DNA or RNA. It shows how the mononucleotides are linked together to form this primary structure. It's just like when amino acids, they come together and form peptide bonds to form oligopeptide unit and then from polypeptide unit. Just like that here, instead of the peptide bond, what we have is the phosphate which is getting linked up with the sugar unit. Now the phosphate links to the sugar unit in the 5 carbon of sugar and 3 third carbon of sugar. So these are the carbon phosphate group can link, and this is how it propagates. So as you can see here, other than the sugar phosphate backbone, the first carbon of the sugar is always linked to the nucleobases using something called glycosidic bond. So this another important feature in case of DNA or RNA, you see that the orientation or the faces of the phosphate and the bases, they are in opposite direction. So one side you have the more ionic phosphate group present, and another side you have in these hydrophobic bases are present. And most importantly, I would like to say this sequence of these bases means sequence of this mononucleotide in this oligonucleotide or the polynucleotide, that is the nucleic acid or DNA, that is very important because that carries the genetic information, and that is what is known as the genetic code. But here we are not interested in knowing more about genetic code, rather we would look into the nano aspect of DNA or RNA. Now if you look at this RNA backbone over here, more or less you get to see from this model that you have just the extra H group at second carbon of the sugar unit in all the cases, and as I said, this OH is very important in hydrolyzing this phosphate group, and that's how alkaline hydrolysis of RNA can happen. Another important difference is that instead of the thymine in RNA, you have uracil group. Now these nucleobases, they can have hydrogen bonding among themselves. So as we see, guanine and cytosine, these nucleobases, they can form 3 hydrogen bonding in between them, and that can be in, if you look at guanine side, it can be donor, acceptor, acceptor. So they complement each other, and it can have 3 hydrogen bonding, whereas in the case of adenine and thymine, you can have a push-pull donor acceptor pair. 2 hydrogen bondings are involved in adenine thymine. Similarly, when we talk about the nucleobases of RNA, so uracil and adenine is involved, you get to see there are 2 hydrogen bondings involved. And now that we have understood the hydrogen bonding pattern between complementary base pairs, let's look at the secondary structure of DNA. So we know also the primary structure where it is the phosphate and sugar backbone that runs particular direction, that is the 5' end to 3' end. 5' means that's where the phosphate group is linked to the fifth carbon of sugar, and 3 prime is that where phosphate group is linked to the third carbon of sugar. And one particular primary structure of DNA runs 5 prime to 3 prime end. Then another strand, they can actually come in opposite direction, 5 prime to 3 prime, and that's where this hydrogen bonding between the base pair can happen in the interior core of this double strand of DNA. And this is very important, that this is what based on Watson-Crick base pairing, and this particular pattern of hydrogen bonding that determines the secondary structures of DNA. Into other important thing is that depending on the segment of DNA you have, which is, it can be AT reached or GC reached. Now we know that the strength of this bonding is different between AT and GC because the number of hydrogen bonding involved are different, and that's what actually reflects when we heat it up, heat this double stranded DNA, so that double stranded DNA melts and hydrogen bond breaks. So that gets clearly reflected in the melting temperature of this DNA. And normally if there is a mismatch of base in between, so that's where the double helix that gets destabilized. That means something is, we can actually understand on the other hand that something is wrong with the sequence in the structure. So let's look at the ribose conformation, or that means it's called also sugar puckering. So there can be two different types of conformation which can be present in ribose sugar. You can see that the carbon adjacent to oxygen is numbered as 2, and in the first case that can be C3 endo. That's where if you take the C3 carbon that is elevated from the plane consisting of the oxygen unit, first carbon and second carbon and fourth carbon. So the C3 carbon is actually elevated, and that's why it's called C3 endo. There can be another possibility where second carbon can be elevated from the plane, and that is called C2 endo. Interestingly, now we know that always C3 and C5, they are linked to phosphate group. So based on these C3 or C2 endo conformation of the sugar, the position, the spacing between phosphate substituents, that actually changes. So most of the DNA conformations which you will see, they are C2 endo conformation, where the distance between the phosphate group are 7 angstrom. But in the other case, there are special cases like aDNA or many cases RNA. We see that ribose sugar stays in the C3 endo conformation, where distance between the phosphate group is 5.9 angstrom. And there is another thing which we will see later when we discuss RNA, is the orientation of this glycosidic bond as compared to the sugar unit, that which can be syn or anti, which we will see later on. The most prevalent secondary structure of DNA what we see in nature under any physiological condition is bDNA. Now in bDNA, it is just like double helix, which is a clockwise helix made from anti-parallel strand. That means one strand is going 5 to 3, another strand is going 3 to 5, and then in between the base pairing happens. Now as we know, phosphate and sugar, they are exposed outside, and the bases are actually in the interior of the DNA, so that they keep mostly the water molecule at bay. And you can see that this one particular complete turn of this DNA helix, that involves 3.4 nanometer of pitch length, and we also see that the diameter of the helix is around 2 nanometer, and which also involves two different kinds of groove sites. One is minor groove or major groove, and as we know, this is where the sugar stays in C2 endo conformation. There can be another structure, secondary structure of DNA, that is called aDNA, but aDNA is more compact secondary structure, and this normally appears at very dehydrating conditions. And what happens is that if you look at this bDNA structure, where your bases, the orientation of bases, they are perpendicular to the propagation axis of the bDNA, but in the case of aDNA, you see they are not in perpendicular relation, rather there is a strong inclination of the bases relative to the helical axis, around 20 degree they get inclined. Another important difference is the ribose conformation here is C3 endo. As you can see, this has really compact structures, and what happens is that the major groove, you see, that becomes very deep. However, the minor groove orientation changes, and minor grooves become very shallow, and since it is very compact, the diameter of the A DNA also increases as compared to the B DNA, and but the the pitch length, as we see from that cartoon, that actually decreases. There can be another secondary structure of DNA possible, which has come much later, that is something called zDNA. And if you look at, again, compare from bDNA and zDNA, first of all, unlike bDNA, where it is a clockwise helix, it is counterclockwise helix in case of zDNA, and this is like a little bit stretched structure. It's just opposite to the aDNA, where it is much more compact. So if you look at the pitch, that is 4.5 in the case of zDNA. However, if you take bDNA, that is 3.4, and aDNA is, as I said, more compact, so it's 2.4 nanometer. And the phosphate backbone is also having a zigzag alignment, and what happens is that it has a minor group, but it doesn't have any major group present because of the zigzag alignment of the phosphate backbone. And as we were discussing, along the glycosidic bond, these purine or pyrimidine, they can have anti or syn orientation, and you can see in the case of zDNA, purines stay in the anti conformation of the nucleosides. However, it is C3 endo for the sugar, but in the case of pyrimidine nucleobases, they exist syn conformation along the glycosidic bond and C2 endo conformation of the sugar. So now we know three different structures of DNA, A, B and zDNA, and you can see on your slide the side view and top down view of the conformation of the secondary structures, and mostly you can see that A and B, where they have right-handed helicity, but Z has left-handed helicity, and you can see that the aDNA is it has a diameter of 2.3 nanometers, so it is little bit diameter is more compared to B and Z, and you can see the aDNA is more compact, but zDNA is more stretched. Normally the formation condition of this different DNA, we have seen A comes in dehydration, and most commonly we get bDNA, but there are some conditions of high salt concentration, we get to see Z form. So we have looked at so far Watson-Crick base pairing, and based on this kind of base pairing, we have seen a formation of these DNA double strands. But there can be also another type of hydrogen bonding, that is something called Hooke-Stern hydrogen bonding, and that is also very important, and that came much later than Watson-Crick hydrogen bonding. And in this case, this Hooke-Stern, you can see there is slight deformation in terms of the bonding angle, and Hooke-Stern hydrogen bonding can be of two types. It can be only Hooke-Stern, or it can be a reverse Hooke-Stern hydrogen bonding, where the atoms that differs on the atoms which are actually involved in the case of purine moieties. Another thing is we need to remember that this Watson-Crick base pairing and Hooke-Stern base pairing, the structures, hydrogen bonded structure which comes out of it, they are in equilibrium to each other. Normally Watson-Crick hydrogen bonding is more favorable and they are more stable, but there are under certain condition Hooke-Stern base pairing gives more stable structures. So here we see, when we talk about Watson-Crick base pairing, we don't demarcate it anything specially, but when we talk about Hooke-Stern base pairing, like you see there is a cross symbol there, so that actually signifies it's a Hooke-Stern base pairing. And you can see on slide there is Watson-Crick base pairing between adenine and thymine, but there is between thymine and adenosine you have the Hooke-Stern base pairing. Now in the case of other structure, you see there is a plus on cytosine, and that has to do with the protonated state of cytosine. That means that's why it has been ascribed like that. This kind of combination of Watson-Crick and Hooke-Stern base pairing is very important for making a DNA triple helix, where you can have a perfect bDNA, and then you can also have another single stranded DNA which can actually come together, where is the red strand which comes together and form it actually 3 strands wind around each other and form this DNA triple helix. And this particular red strand that actually binds to the major groove of double stranded DNA by Hookstein or reverse Hookstein hydrogen bond and form triple helix. In this case, we can see not only triple helix, we can see that Hookstein pairing is very important in forming other kind of DNA secondary structure, something like G tetrad and G quadruplex. So what happens here is you can see that there are 4 pyrimidine base pair. These are all G guanine base, and they can form Hookstein base pair among themselves, and 4 guanine can come together. That's why it's called tetrad structure. And in the down we have shown 3 different possibilities of this tetrad. So now what happens is that now this tetrad would have one plane, and the different tetrads can come in the stacking plane and form G quadruplex. So the first example where you have G quadruplex, where it is involve 4 DNA strands in parallel arrangement form this G quadruplex. The second type of G quadruplex, where you can have 2 DNA strands come together then they can form intermolecular G quadruplex. Also, there are examples where you have a very long polynucleotide DNA chain, and that can actually come and form anti-parallel but intramolecular G quadruplex. And this shows a long polynucleotide chain where you have different region, where you have this different loop region and different region where you have guanine rich sequences. And what happens is that regarding the loop region, all this G, they come together and form very nice tetrad, and eventually they form this quadruplex moiety. Normally, in order to stabilize such a kind of stacked quadruplex moiety, you need to have a stabilizing unit. Mostly it is cation at the center which stabilizes this kind of G quadruplex structure. In this slide, we see different possible binding sites in DNA. So as we have mentioned earlier, a double-stranded DNA can have a major groove, or it can have another side of the major groove, that is minor groove. And we must remember that DNA has sugar phosphate backbone which is exposed to outside water, but the base pair, that is hydrophobic in nature, they are actually buried inside. So there can be electrostatic binding to the sugar phosphate backbone, so which is a negative charge. So many cation positively charged ion can get attracted to DNA by electrostatic interaction. Group binding, there can be particular molecule like distamycin or ethylhistamycin, such a family of compounds that actually can bind to the major groups or minor groups depending on the ligand fitting. And there is also another mode, the third mode of binding to DNA, when you have a large pi electron surface, and the base, as we know, we have the base pairs which also have large pi electron surface. So like compound like ethidium bromide, they can actually go deep inside and it can intercalate with the base pairs. So after we have got to know about DNA, let's look at the structure of RNA. So what is RNA? So RNA is ribonucleic acid, and it's again a polymeric molecule, and it has roles in it has a lot of biological roles in coding, decoding, regulation and expression of genes. There are mainly 3 types of RNA. One is messenger RNA or mRNA we call, that actually conveys the genetic information. Then other type of RNA is transfer RNA or tRNA. These tRNA molecules they deliver the amino acid to ribosome during the protein synthesis pathway. And the third type is ribosomal RNA or rRNA. They actually link those amino acids together to the protein. So RNA actually works in tandem with DNA in terms of protein synthesis, and also many for transferring genetic information. However, unlike the double-stranded DNA, RNA is normally single-stranded molecule, and RNA can have a complementary base pairing, even though it doesn't form a double-stranded structure. But this base pairing can be intra-strand. That means in the single strand of RNA, they can come and they can form this helical structure. Just like this is more prevalent in tRNA, we'll see that those structure as we go on. However, basic two difference, as I again mentioned, I am going to mention, so where DNA, the sugar unit, has deoxyribose, but RNA contain ribose, and that means in ribose and deoxyribose, the difference is between that two hydroxyl group at the second carbon atom in the sugar ring, and this hydroxyl makes RNA less stable towards alkaline hydrolysis. And another difference, that is the complementary base to adenine in DNA is thymine, but in the case of RNA, the complementary base is uracil. Uracil is nothing but an unmethylated form of thymine. There is a very close similarity between uracil and thymine. So now let's look at the hierarchical structures of RNA. So we have already know about primary structure of RNA, where you have Watson-Crick base pairing, but you have ribose sugar present here, and the presence of uracil instead of thymine. Now, this primary structure of RNA, there can be Watson-Crick base pairing, but it doesn't have any double-stranded RNA presence. Rather, RNA stays in single-stranded form. But what happens is that there can be intramolecular, intra-strand Watson-Crick hydrogen bonding, and it can form secondary structure like what you see. It can form something like stem, or it can form loop region through this intramolecular, intra-strand hydrogen bonding, which can also lead to tertiary structure of RNA in 3 dimension. If you look at more carefully the secondary structure here, you can see when you have complementary structure of AU or CG, and then you have perfect Watson-Crick pair, but there can be also mismatch, like you have both the strand EU, so then that's where that will actually bulge out. So that will so there is a stem formation due to mismatch, there is something bulge formation, and there can be also loop in that. So and there can be different types of loops or bulge region, as you see from the secondary structures of RNA. So let's summarize this module. What we have learned so far in the module. So we have looked at more detail about the structure of DNA and RNA. First of all, in DNA, what did we see? We first of all looked at the molecular level. What are the molecules DNA or RNA made up with? We have looked at sugar unit, phosphate backbone, the different bases. Now we looked at the primary structure, how 5 to 3 strand, and how is the antiparallel orientation, it actually forms the secondary structure. So this is important. We learned the secondary structure formation, and how is the difference between Watson-Crick base pair if you are doing it between G and C or A and T, and like how is how during this hybridization process the percentage of AT or GC, it determines the melting of this double-stranded DNA. So those are the things we learned. And different kind of structure, double helix structure of DNA, in which condition, like A DNA, B DNA, Z DNA. Now, which particular conformation of DNA or structure of DNA actually expresses more in which particular conditions, like dehydration condition or normal physiological condition or very high salt concentration. So that we got to know. We have also looked at the different structural difference between these different parts of DNA, different conformations of DNA, and then we have looked at also the RNA structures and how RNA can form this intra-strand base pairing. And lastly, we have also seen that there is not only Watson-Crick base pairing, there can be also something called Hook-Stein base pairing, that can be reverse Hook-Stein or normal Hook-Stein base pairing, and that can also give different structure in DNA. So it can have G quadruplex structure or triple helix structure of DNA. And lastly, we have looked at possible binding sites in the nucleic acid, and based on that it will help in designing drug. So if you want to do targeting, like if you want to do electrostatic interaction, or you want to go for minor group or major group, or you want to go for an intercalation mode. So depending on your drug and looking at the molecular structure of the drug, you can actually say that where it should go and which is the potential site for binding. So I hope it will be helpful for you to understand the strategy to design nanostructures. Thank you.